Good morning, or good afternoon, everyone, and welcome to LifeSciences BC Showcase Series, Vancouver Island. Wonderful that you're able to join us today. We've actually have record attendance of people who are able to attend. So this is great. And for those of you who don't know, my name is Ryan Budden. I'm the Director of Partnerships and Events with LifeSciences BC. And we are very excited to highlight all the wonderful things going on in Vancouver Island. So just a couple of features before we get started. You will see the chat function there, so you can participate throughout, put any questions or thoughts or anything like that during the event. For questions specifically, we ask you to put that in the Q&A. There's not going to be a specific Q&A session at the end, but throughout, all of the speakers will go in and make sure that they can answer your questions there. We also encourage you at the end of the event, we're going to have some networking, and therefore you will be able to meet all of our speakers and chat with them as well. One thing also in the Q&A that's great, we have an upvote function, which is great. And that then allows you, if you see a question that you really like, it goes to the top and our speakers are able to see that. At the end, we're going to have probably, depending on time, 2 to 3 rounds of networking, which will be around 8 minutes each. I encourage you all to move to the front of the room so that you're able to meet with all of the speakers and then fill out the rest of the area. To move from table to table, you hover over a table, double click on that, and then encourage everybody to introduce yourself for a minute and then have a conversation for about 8 minutes. We would ask that you please wait until I announce the break before you move to another table, just so that it's not disruptive for the people having the conversations. Also a reminder, when you're at the tables, to please turn your cameras and microphones on so that people are able to see you. And if you've not had a chance to do so already, please upload your photo and your social media links. So to get us started now, I'd like to introduce Wendy Herbert, who's our President and CEO, and she's going to be your MC and introduce everyone today. So over to you, Wendy.
Okay, so welcome everyone to our 4th annual LifeSciences BC Showcase Series. And today we are focusing on Vancouver Island, which we are super excited about. This day is, this event is very memorable to me because exactly a year ago, we were sitting in Victoria at the Delta Hotel hosting this event where 100 people gathered to learn about innovation and research happening in Vancouver Island. It also marks our last in-person event that LifeSciences BC hosted. So quite amazing. You know, in some ways the year has flown by, and in other ways it has, you know, it's gone quite slowly, but it's really interesting that now we are here virtually doing this. And while we're sorry to not be in Victoria, I love Victoria, so I'd love to be there. But one of the advantages of it being a virtual event is that we can actually showcase the amazing research and innovation that's happening on Vancouver Island to a much broader audience. And we have over 200 people that have registered for this event. So clearly there is a lot of interest in this. I want to introduce Scott Phillips, who is the President and CEO of Starfish Medical, and also the Chair of the Board of LifeSciences BC, to say a few words before we could begin.
Great, thank you, Wendy. Well, I won't take up too much time, but I think it's actually quite nice to be have LSBC have enough representation around the province that the chair can be from Victoria here on Vancouver Island, and I often talk about critical mass in the industry and how do we get to that spot, and seeing some interesting things on the island is super impressive this year. I think that for the first time ever, there's boxes of drugs shipping all over the world and that actually have a Victoria address on them with Aurinia Pharma. That's a major achievement that we can all be proud of. And you'll hear from some others. Hopefully there'll be some more stories following the footsteps of that story. So if you don't know much about LSBC, we're a nonprofit. We bring together the voices of the technology community itself, universities, educational institutions, other nonprofits. Just how do we bring the voices together to have our whole industry become successful together? So if that's what you're interested in, you're in the right spot, and you'll hear a lot of things that support that in today's event. And I always emphasize the community piece. Somehow, what's going to allow us to be successful here is people supporting each other. And this is such a wonderful industry that way, because none of us really compete with each other. How can we support each other to find ways to be successful? And I think you'll find across the industry that people are pretty open to having conversations and helping you support your new companies and so on. So I hope you have a wonderful time today in this session, and I will pass back over to whoever's next on the agenda.
Thank you very much for those words. As we begin, I would like to acknowledge that I'm joining this event from within the ancestral, traditional, and unceded territory of the Coast Salish peoples, including the territories of the Musqueam, Squamish, and Tsleil-Waututh First Nations. I'd also like to acknowledge that our other speakers are joining us from other traditional and unceded territories across this beautiful province. At every event, we like to welcome our new members. So our latest new members are Hamilton Company, Herstasis Health, and Satisfy Health. So wonderful to have you all as new members. We are always welcoming new members, and please reach out to any of us at LSBC if you're interested in membership. It's actually our annual renewal of membership time, so there's quite a bit of activity going on with that. So with that, I want to turn to our first speaker, David, who is all lined up here, which is great. David is the CEO of Oak Bay Biosciences, a Victoria startup formed in 2020 to advance a portfolio of aptamer-based assets for therapy of retinal disease. The lead molecule blocks complement factor D activity and is intended as a therapy for Stargardt disease and other forms of progressive retinal degeneration. Prior to joining Oak Bay Bio, David served as a director of R&D for Overa Inc. and director of laboratory services at ImmunoPrecise, another Victoria-based company. David completed his postdoctoral training with Brad Nelson at BC Cancer Agency's Dealey Research Center, who actually spoke at this event last year, after earning a PhD in immunology at the University of Saskatchewan. In addition, David is one of the recent first grads of our investor readiness program that we just completed yesterday. So over to you, David.
Sounds good. Well, thanks, Wendy. And I guess I don't have to introduce myself too much. I was just thinking back, this event was the last event that I attended in March 2020. And I was thinking about how nice it would be to sit on the patio at the Ocean Point this afternoon and have a beer, because we have fabulous weather here in Victoria. And so it's great that I can, tell everybody, introduce Oak Bay Biosciences. We're a fairly new company in Victoria. And we're founded on an acquired portfolio of assets, and we're aimed at developing a therapy for Stargardt disease. So I thought maybe I'd start by introducing the team, because it's, this is a Vancouver Island showcase. So I wanted to just mention that most of us are connected to Victoria in some way. So I'm about a fourth generation Victorian. And the founders and board of directors, Eric Moore and Paul Erickson, are longtime Victoria residents as well. You may recognize Paul and Eric from different companies around Victoria. And of course, we've got some advisors in the city as well, not least of which is Sam Mercer. So we're well connected in this community. And speaking of which, if you come to Victoria and you look across Cabarrot Bay towards Samuel Point, you might see something like this. If you had Stargardt disease, though, you may see something like this, which is basically a black hole in the center of your vision. And that's bad enough. But if you wait long enough, unfortunately, you would progressively lose that little vision that you have and eventually become legally blind. So there aren't any currently approved therapies for Stargardt disease. It's an inherited disease. It affects about 800,000 people worldwide. And it leads to progressive and irreversible blindness, and often beginning in childhood. And this blindness is caused by accumulation of toxic bisretinoid vitamin A compounds that accumulate in the retina. And this causes cellular stress and activates a component of the innate immune system called complement. And this eventually leads to death of the retinal cells in the macula. You can see in the right-hand picture of the retina of a Stargardt patient is essentially an area of dead retina that leads to that blind spot that I mentioned. So fortunately, because complement is strongly implicated in the pathogenesis of the disease, preventing the activation of complement can lead to about a 90% protection of cellular death in the mouse models. And this is actually a mouse model published by one of our advisors at UCLA. Inhibiting complement rescues the phenotype in the ABCA4 knockout mice. And that's important to us because our lead molecule is an aptamer, inhibits complement factor D. Factor D is a clinically validated target that's a central part of the activation, an early part of the activation of alternative complement pathway, which is the main pathway that leads to damage in this disease. And the molecule and our company have been extensively de-risked prior to the acquisition by Biosciences. So we estimate about 4 years and $5 million was invested in development to date. We have 2 issued patent families. The drug is the same drug class as Macugen, which is an aptamer that's approved for AMD. The molecule has gone through extensive optimization for potency and durability. We've completed multiple PK studies in rabbit models, and also have had successful manufacturing and scale up. So what we're doing right now is to complete a couple of further de-risking activities to complete the preclinical data package before we enter the path to clinic. And so we're collaborating with 2 local groups. The first project here is to formally demonstrate a role for factor D in the Stargardt model, and thereby further de-risk this target. And we recently received MITAX funding for an industrial postdoc, Bridget Ryan, who came out of Bob Chow's lab at UVic, and she'll be staying on for the foreseeable future to help us with these studies. The mice in this experiment have been ordered. They're actually potentially playing mood music for them right now as part of their mating. And our second activity is to demonstrate lead molecule activity in human Stargardt serum. So it's basically to show that our molecule can inhibit complement in patient-derived specimens. So either one of these activities could lead us to orphan drug designation, and that is our main goal right now. So we think with a fairly ambitious timeline, running the mouse model will take some time, a little bit over a year. We could achieve orphan drug status within 18 months. And during that time, we're building our scientific advisory board. I can tell you more about them if you want to chat, engaging KOLs and building our team. We're building the team particularly because after this positive data, we'll be going to be raising a Series A round of approximately 20 million that will allow us to enter the path to clinic studies. So we'll be doing clinical talks and also scale up GMP manufacturing. So currently we're raising $1 million angel round, and it's about 40% full. So yeah, I would be, not doing my job if I didn't mention this, but as I said, we've got about 380,000 committed from angel investors and founders. We're offering favorable terms, and be happy to chat with you more about that. And finally, I just wanted to leave you with my contact info in case we're not able to connect. We have a website we recently launched, oakbaybio.com, and you can follow us on Twitter. Thanks.
Thanks very much, David. Great presentation. And again, congratulations on completing the investor readiness program. Our next speaker is Dr. Chinetta. Well, I'm going to introduce Dr. Chinetta Jones from the Michael Smith Foundation for Health Research to introduce our next speaker, Dr. Shannon Dames. David, you can turn your camera off. Hi, Chinetta. So as VP Research, Chinetta Jones leads Michael Smith Foundation for Health Research funding programs, ensuring support for our province's talented health researchers in improving the health of British Columbians and their health system. She also guides our knowledge translation, partnership, and evaluation activities to maximize the impact of Michael Smith Foundation's investments in health research. Chinetta has more than 20 years of experience in science research strategy and evaluation of government and philanthropic sectors in the US, UK, and Europe. Most recently, she was Director of Insight and Analysis at the Wellcome Trust in London, where she provided strategic leadership to maximize the impact of the achievements and opportunities of their funded research initiatives. Chinetta is an active champion of equality, diversity, and inclusion, and has held a long-standing commitment to mentorship of emerging leaders. Over to you, Chinetta.
Thank you, Wendy, and thank you for the kind introduction. Hello, everyone. Greetings from my home in Vancouver on the ancestral and unceded territory of the Coast Salish peoples, including the Musqueam, Squamish, and Tsleil-Waututh peoples. I want to express my congratulations to LifeSciences BC and my deep thanks for organizing a fantastic series and for today's event that highlights the vibrant research in life sciences at Vancouver Island. I'm honored to be here on behalf of the Michael Smith Foundation for Health Research. For those of you who may not know us, we are British Columbia's health research funding agency. We are funded by the government, and the core of our work is to support health research talent in BC. We help to attract, retain, and develop talent that will advance science and health research and move research evidence into action for the benefit of all people and communities in British Columbia. And I'm thrilled to share a new funding opportunity that we just announced today. We have partnered with Genome BC and the BC CDC Foundation for Public Health to fund research that addresses critical questions around the COVID-19 vaccines, their performance, access, and public perception. So please do see our website for more information about this funding opportunity. And after my brief words, I'll put the link in the chat. As a provincial health research funding agency, we recognize the importance of fostering health research capacity in all regions of the province. It's essential that we support research that is important to local communities, that we support local researchers, and that we work in partnership to strengthen local health research capacity in every region across the province. Since 2001, the Michael Smith Foundation has invested $27 million through 152 awards to Vancouver Island researchers based at the University of Victoria, Vancouver Island University, Royal Roads University, and Island Health Authority. And we are extremely grateful to community organizations like the Victoria Hospitals Foundation and the Hecht Memorial Foundation that partner with us on funding awards to build regional research capacity. But we know we can do more. We want to increase the regional diversity of our funding programs and to fund more researchers across Vancouver Island. So over the last several months, we've consulted with our regional colleagues and partners to implement improvements to our funding programs to improve the success of regional applicants. And on that note, it's my pleasure to introduce a Michael Smith Foundation Health Professional Investigator, Dr. Shannon Dames, who will speak on revolutionizing mental health care with psychedelic-assisted therapies. Dr. Shannon Dames comes to the Roots to Thrive ketamine-assisted therapy team with a focus on research and development, ensuring program methods are evidence and patient-informed. She developed the Roots theory and works as a nursing professor and resilience researcher. As a byproduct of her doctoral work that focused on the core factors that promote human flourishing, and as a health professional investigator for the Michael Smith Foundation for Health Research, Dr. Dames is now spearheading the ongoing development of the Roots to Thrive program and the integration of psychedelic-assisted therapies. It's my pleasure to hand over to Dr. Dames.
Yes, thank you for the introduction. I apologize for that mouthful. My name is Shannon Dames, and I am delighted to be here on this very exciting topic. The first screen or the first slide, which is now missing, but it's I just really want to acknowledge that I am able to continue this work that I've developed because of the Michael Smith Foundation for Health Research. And I am very fortunate to be a receiver of the funding that enables me to kind of take this in a much broader way than we would have been able to without it. And just to give you a little bit of context, the funding that we received is really around a publicly embedded and offered psychedelic-assisted therapy program. So, and the purpose of it is not to build one program, but it is to build a roadmap. So essentially what we did is we took a resilience development program, which has been developed by a multidisciplinary group of caregivers and experts. It is an open access program. It was developed in conjunction with a health authority and an academic institution, and has had some pretty positive feedback. It's specifically aiming at healthcare providers. However, we've also offered it to the public as well. So we took a community of practice and we combined it with ketamine-assisted therapy, which is 3 sessions over a 12-week program to see if we could really work with trauma in a new way and essentially to take people a little bit deeper than we could at the level of the mind. So, and I'll talk a little bit about how these psychedelic-assisted therapies work with trauma and healing, and really enabling people to kind of feel into their kind of best, most resource self. So with this, we've done a 3-prong approach. The service delivery arm, we developed a container that could hold the services and in a safe way, which requires a whole heck of a lot of work to create something that feels secure enough that people can let their guard down in. And then we are now, because of the funding that we've received from the Michael Smith Foundation for Health Research, we are able to research that program through a formal harmonized review board, which is what we're in the process right now. And we are also developing a parallel graduate certificate for those that would like to do this advanced practice, which would be the first in Canada in an accredited 4-credit program. So our first pilot that we did, which was funded through the Canadian Institute for Health Research, and we ran 16 participants through. We had 17. One had to withdraw early on before any of the ketamine sessions. So we had 16 that went through from start to finish. Weekly attendance was very high. People only missed for urgencies and emergencies. Most people were there every single week for 12 weeks. We had, sorry, I have these on a timer to keep me going. Our results were substantially positive. We were all actually a bit surprised at how positive they are. We had 11 come in screening positive PTSD. All of them left negative. Generalized anxiety, also quite high. Depression, significant improvements. 54% now screening negative after moderate and severe scores coming in. And self-compassion rates were all substantial as well, with those coming in with low self-compassion, leaving with moderate or high. And we also saw significant improvements across the work-life functionality scales. So what we did to make all this happen is we pulled in a group of experts from, really, this is a handful of them, and there are many more that have leaned into the Roots to Thrive program, which is the container holding all of this work, or the ketamine-assisted therapy component from across the province, even really across the country, have kind of leaned in and provided expertise. This group here is kind of the one that's holding the container on the week-to-week basis. We now have 36 patient partners that are now in the program kind of as we speak, meeting on a weekly basis. So why did we do this? We did this because we know that we're in a mental health crisis. And I think most of us all know that we're in a mental health crisis at this point, and not just because the statistics are staggering, but because we are now no longer others. Well, I'll start with this slide here. We know that even with those receiving treatments, maybe up to 4 million are served by the traditional mental health system, but we know that there is another almost 15 million, likely more now, post-pandemic, that are not being served by the traditional therapies. So adding more to this, we, by the time we reach the age of 40, 1 out of every 2 of us is diagnosed with a mental health condition. This is no longer something that's happening to other people. This is happening to us, within our communities, and we are seeing a much higher demand for treatment methodologies that are more effective. So we also know that suicide rates are higher than ever in many areas, and that our current mental health toolbox is failing really for most people. So we're looking for not doing away with the old system. We need that old system. It works for a lot of people, but really expanding the mental health toolbox so that we can reach an even wider audience. And not to do this flippantly, but to do it in a way that is incredibly well vetted and safe, both psychologically, culturally, spiritually, with people that are, showing up with their own kind of authentic ways of being and wellness in good order, so we can create a secure environment for people to heal in. So RTT CAT is basically what we've done is it is a recognition that we do not heal in isolation. Even in the traditional psychotherapy environment is not necessarily working for many people as we had hoped. So we are really modeling a system, we're really piloting a model that focuses on community as the core methodology for healing. And within that community, people learn and experience secure attachment to each other, and they can then direct that inwardly. So we do that through the medicine in this. While it sounds a lot like it's, whether it's ketamine or other psychedelic producing medicines, the medicine that we use in this program is actually unconditional positive regard, felt through connection, which then promotes the secure attachment necessary for us to heal. So basically what we do is we take people in through a suitability assessment. We really make sure that we're aligned. We do a medical intake, which is actually 3 phases. It's very, very thorough. And this is prioritizing safety across a holistic safety, really, both psychological, biological, et cetera. We match people up in a community of practice, which is consistent throughout the 12 weeks. These people also, the participants, sit within that community of practice for their ketamine sessions as well. So these are not done on a one-to-one basis. We do the psychedelic assisted therapy sessions in their community of practice groups. So they're sitting in groups, COVID friendly. And then we do integration sessions ongoing throughout the 12 weeks, where we're really digging into helping people kind of show up vulnerably, see each other, witness each other, be in a container of unconditional positive regard as they do their healing work. So we do all of this as a bit of a layering technique. We know that connection is key, and having an ability for people to test the container and start to develop trust and relationship is how people deepen in that connection. We also know that trauma we consider it, these stuck energies that we can't always access at the level of the mind. And we use these psychedelic, meaning mind loosening, mind manifesting sessions to kind of work around the nervous system and the ego so that people can get to those kind of stuck energies and release them in a way they couldn't on a level of psychotherapy. So what are we doing now? The goal at this point and our whole purpose, we are a public Vancouver Island University. We have received funds only through grant funding. We really have no conflicts of interest apart from that. Our goal is to demonstrate that we can do this in the public system, that we do not need to have the corporations come in and build this for us and have a kind of 2-tiered medical system. We're in a mental health crisis. We need all hands on deck, and this cannot just be a private venture. So our whole goal is to embed as best we can in the public infrastructure and work out all of the kinks to do that, and to help kind of pave the path for others to do the same, because we know that the need is absolutely emergent at this point. So yeah, I think that's all I've got. I feel like I've run out of time.
Thank you very much. That was super interesting. I look forward to connecting with you after to learn more about it. But yeah, very, very interesting. And if you have questions for For Shannon, she will be available during the network. Source her out. Our next speaker is Dr. David Goodlett. So David, if you can turn on your camera. Dr. Goodlett's 30-year career in mass spectrometry to solve biomedical problems via novel technology and software developments led him to the University of Victoria, where he's a visiting professor and holds the Dawn and Eleanor Ricks BC Leadership Chair in Biomedical and Environmental Proteomics. He's the director of the internationally acclaimed Genome BC Proteomic Center. His research interests lie in developing the lipid A structure-activity relationship in order to elucidate the basis by which lipid A acts as agonist and antagonist on binding MD2/TLR4. He has two startups, Durian.com and Patagen, I may have pronounced that wrong, .com, focused on MS uses in the life sciences. He is a co-organizer of msbm.org, a mass spectrometry summer school, and is an editor at Rapid Communications in Mass Spectrometry. Over to you, Dr. Goodlett.
Right, so I'm Dave Goodlett. I'm a professor in the Department of Biochemistry and Microbiology, and I'm director of the Genome BC Proteomic Center here at the University of Victoria. I'm gonna, because I've got two hats that I wear at least, I'm gonna tell a little bit about both positions, and we'll start out with a few slides just as an overview of the Proteomic Center. So the center's been around for about 30 years, and they do, or we do, targeted and discovery-based metabolomics and proteomics using mass spectrometry. With metabolomics, it's a little more straightforward. With the proteins, we have to digest those to peptides. But this is a complicated slide, but the short story is that we can tell you what's in your sample, and we can also tell you how much is in your sample. And that's what you sort of see in this middle panel here on the far right. We can quantify changes as a function of perturbations by spiking in standards that are heavy labeled with, say, C13. Okay, so there's lots of omics that we do as fee-for-service, but also research. We do a bit of protein characterization as well, so various kinds of techniques like chemical cross-linking to do structure analysis, small molecule structure analysis as well. The rub here is that these projects, structure projects, are a little more difficult than just the omics projects. And so as a result, they cost a little more and take a little bit more time, but we're capable of doing those kinds of things for you. And then last slide about the center. As a result of generating lots of different kinds of data, we also have quite a bit of informatics support to help with report writing or patent filing, whatever you're doing, where you need to explain what's been done at the center. And if you have any questions, you can Google UVic Proteome Center. I think you'll find us, and you can contact me or Dr. Dave Shibley. Okay, so over to a research theme that the lab's working on, and it involves lipid A, which was alluded to in the intro. And the short story is we're using various kinds of microbial membrane lipids to identify different microbes. So gram-negatives, of course, have lipid A as an anchor in their outer membrane. And there's quite a bit of variation in these lipid A molecules. As was said in the intro, sometimes it can be an agonist, sometimes it can be an antagonist. Gram-positives have different molecules in their outer membrane, and then fungi, yet again, have a different array of molecules. And all of these, it's a story for another day, but all of these lipids can have biological activity. So what you see here are, on the left-hand side, 6 panels of mass spectra, A through F there. And basically, we go through a simple extraction protocol using this is MALDI-TOF mass spectrometry. So on the right-hand side here, you can see where it says apply sample. We basically pipette the sample down, do a little bit of chemistry, and then put the plate in the mass spectrometer, and then we generate these spectra that are unique to a given organism. So like panel B here at top middle, Klebsiella pneumoniae, always shows this ion at 1840. Pseudomonas aeruginosa always shows this ion at 1446. And there's a number of papers here that we published recently explaining how this method works. But basically, we have a process that looks a little bit like the Bruker biotyper, which is a protein-based process. In our case, we're using lipids. We don't need to know, is it gram-positive, gram-negative, or a fungi to start with. We just put these into the mass spectrometer, generate a mass spectrum, and then we've got this lipid barcode that we can read out to give us an identity. And you might say, okay, well, there's the Bruker biotyper. Why do you need another form of microbial ID that uses mass spectrometry? And in our case, we've got one up on the biotyper in the sense that we're able to easily identify multiple microbes together in a sample. So this means we can do mixtures, which you would find in biological fluids like urine or blood, as you'll see in a second. The Bruker biotyper requires that you pick a pure colony off a plate. So we in essence cut a day off of the process of making an identification because we can skip growth on the plate. We can basically just take a sample of urine, process it, and generate spectra. So that was 3 organisms acquired together. Here's 6 different panels. This is a bit contrived, but each one of these, from C. albicans over to Klebsiella pneumoniae, was grown in urine, and then we extracted a microliter of urine, put it on that moldy plate, and generated these spectra. What I don't have to share with you is an actual sample, but we do have some nice data that we've produced recently where we are working with a physician at the University of Maryland, where I came from before I arrived here. We're working with the clinical diagnostic lab there to identify bacteria in urine and basically to call urines as positives or negatives. And it turns out that about half the time that somebody thinks they have a urinary tract infection, it's negative. And so physicians really need a faster way to move on from the patients who think that they have a problem that's related to an organism like a gram-negative bacteria, when in fact it's not. And so we're able to do that a day faster than the standard process. And we've started to work with or have discussions with Michael Chin of Island Health to use a cohort here in British Columbia to compare to what's going on there in Baltimore City. And you can see this little logo at the bottom. There's actually a spin-out company that was mentioned, Patagon, in Baltimore and Seattle, that's commercializing this process. Here on the left, you can see this blood bottle. So if you go in the hospital and the physician thinks that you're developing sepsis, they're going to take some of your blood, put it in this blood bottle, and grow it till it gets to be about 10 to the 9 bacteria. Well, we only need it to get to about 10 to the 5 bacteria, and we don't need to plate it. So if you look at these 2 different flows, the conventional blood culture pathway requires this subculturing. So you take it out of the blood bottle, you plate it on a Petri dish, and it grows overnight or 24 hours at least. In our case, we can skip that step, so we can actually do a little bit faster job of determining what the organism is and whether or not it's susceptible to a given set of antibiotics. Okay, and just one more final thing to sort of switch tones a bit. This is a project that's funded by Genome British Columbia. So with proteins and metabolites, we don't have polymerase chain reaction, PCR. So whatever we start with is what we have to work with. And every time you pipette a solution of peptides or proteins or metabolites, you lose some. You just don't know which ones you're losing. And so the whole game with trying to work with small amounts of sample really depends on minimizing adsorptive losses to surfaces. And so a colleague of mine who's now at Brigham Young University developed this process that basically uses nanoliter-sized wells. And so you put your proteins in and basically never pipette again. All the chemistry happens in this nano well, which you can see at the bottom and the middle part of this slide with this, I think that's an American quarter there. But it's basically a glass slide that you do all your chemistry in. Once that's done, then you pull it out and go straight into the mass spectrometer and do things as you would normally do. And so one of the members out at the Proteome Center, Azad, is working on this process. We're hoping that Anuj, who's the new postdoc working in PETCH, will be able to use something similar to try to identify bacteria from, if not single cells, 10, 100, or 1,000 cells. Okay, so that's it. I want to say thanks to Bob Ernst, who's at the University of Maryland, but I met him 20 years ago when we were both at the University of Washington. He's my microbiology collaborator for 20 years now. And the folks on the right-hand side are the new team of folks. A couple at the bottom here are out at the Proteome Center, and the others are working in and out of PETCH. And thanks to the NIH that funded the diagnostic project, Genome BC for the SIP single cell proteomic project. And then this was also mentioned in the intro. I just want to say we're probably not going to be able to have this meeting this year because of COVID and the slow vaccine rollout. But next year, I think we'll be on again. This is an annual event that takes place over the course of a week in Dubrovnik, Croatia, where you learn fundamentals and applications in mass spectrometry. Okay, thanks very much.
Thank you very much. Very.
Let's see.
Thank you very much. Super interesting presentation, and loved your second last slide with the silhouette of the buildings. Our next speaker is Dr. David Llewellyn. David is the Chief Commercial Officer at Stem Cell Technologies, Canada's largest biotech company. David is a champion of stem cells' mandate to advance science, which he supports at the executive level by ensuring the company is well positioned for long-term growth and profitability. David started his career at Stem Cell in 2006 as a business development officer. Today, he oversees the company's marketing, communications department, pricing, e-commerce, and several other commercial activities, as he was recently named the Chief Commercial Officer. David has a BSc in chemistry from the University of Northern British Columbia, a PhD in organic chemistry from McGill, and an MBA in strategic management from the University of British Columbia. He's a very passionate leader of the life sciences sector, so we're so pleased to welcome you once again at one of the showcase series and appreciate Stem Cell's support for them. David's also on the board of LifeSciences BC. So over to you, David.
Just as a bit of a background for those of you that are not as familiar with stem cell technology. So we are a privately held company. We don't have any investors. We're not a public company either. And so, everything that we do and all the work that we do in our growth is funded through the revenue that we generate from the sales of our products. So we're a profitable company. We have about 1,400 employees, the majority of which are here in the Lower Mainland. We typically grow at about 20% per year. And like I said, head office is in Vancouver, and this is where about 85% of our operations are. So on the right-hand side here, just have a few of our buildings, just some examples. This 1618 Station Street, this is our research and development center. It's right by Science World. The 570 West 7th Avenue, that is by the BC Cancer Agency. That's one of our manufacturing facilities. 750 West Pender, this is our sales office. And then the 8255 North Fraser Way, that's our distribution center in Vancouver. And then Production Way is a new site that we're actually just building out right now. So there's only a couple hundred people at this site, but there's everything, we're going to have a lot of our operations are going to move out to Production Way. I'm going to talk a little bit more about that later on in the presentation. So I wanted to start just by going back about 40 years. And if you think about how we treated disease, before the 1980s, it was largely with small molecules. That's changed quite a bit over the years. In the 1980s, you had the introduction of biologics, antibodies and things like that. And then now we're seeing another transition that's happening right now, and that's this transition to cell therapies. And if you look from that shift from small molecules to biologics to cell therapies, that shift and those advancements are driven largely by the underlying tools that are available to the people that are developing these therapies. So stem cell, we don't do any drug development ourselves. what we do is we develop the products that are used to develop the drugs. We either develop the products that are used to develop and manufacture the biologics, and the ones that are used to develop and manufacture the therapies. And if you look at our portfolio of products, we have about 2,500 products, I believe, on the market right now. And they fall into kind of 4 distinct categories. They're cell sourcing, so products to help researchers access the cells or the cells themselves that they need, like leukopaks and blood. We have products to purify specific cell types from the blood or whatever the cell source is. Once you have the purified cells, we have products to culture them, and then we have products to do the downstream analysis. And so our major product lines are cell separation reagents, cell culture agents. We sell things like cytokines, small molecules, antibodies, those sorts of things. We have CRISPR-Cas9 gene editing tools, and more and more instrumentation coming out. And we also do contract assay services for our customers as well. So very focused on the science and very focused on the workflow in these areas. So just as an example of one of our product lines, this is our EasySet product line. So I thought I'd kind of just dig in a little bit in terms of how the technologies work, so you get a feel for the types of products that we sell. This is, if you're, say, for example, a COVID researcher and you want to look at the immune response for someone who's had COVID and who has recovered, what you might want to do is you might want to study their T cells. And so you take a sample of blood, and then in order to isolate the T cells out of that blood, you would use our EasySet product. And the way that this works is that if you look at the orange part here that I'm highlighting, that's an antibody that goes after some sort of protein on the surface of the T cell that's specific for the T cell. So only the T cell will have that protein. The antibody is specific to that protein, so it kind of binds onto it, and then that antibody to the T cell is cross-linked through 2 other antibodies to another antibody that is bound to a magnetic particle. So you basically cross-link the T cell to a magnetic particle, you kind of coat it in magnetic particles, and then over here, you put it on a very strong magnet, and then you can pour off the cells that don't have the antibody, and what you're left with in the end is just your purified population of T cells or whatever. So you can do it for pretty much whatever cell type you want. So a very good, easy, efficient way to isolate very, very highly pure populations of cells in about 15 minutes. So we have a series of magnets for people that want to do it by hand. And for people who want to automate it, again, we have our instrumentation division, and we have a series of instruments that allow you to automate the process for people who need a higher volume. In terms of our distribution, so like I said, most of our operations are in Vancouver. We do have operations in the US. We have a sales and marketing and some training in Boston. We have a distribution center in Seattle. In the UK, we have a distribution and a sales center, and we also have a research lab in the UK now. Distribution and sales in Germany, distribution and sales in France, and then distribution and sales in 2 locations in China, as well as Singapore and Australia. So you can see the part marked in orange. These are the major markets where we serve directly through our sales force, a very highly trained sales force. And then in the dark gray, or in the dark gray, this is where we sell through distributors. So we sell through distributors in markets where we haven't kind of reached a point where they're large enough markets to set up our own office. And so in those cases, we sell through distributors. And I think a big part of what we've been able to do, it's not just the capabilities to do the sales and marketing in these countries, it's also the logistics and the supply chain that we have set up. It's not always easy to import and sell regulated products in the various countries. Each country has its own set of regulations, so you have to be very aware of what you can and cannot import, and where you can sell, say, an IVD or a GMP product, and make sure you're compliant. So we have a very good network, distribution network, to make sure that we're compliant and we can do that importation. So just going back to that production wave facility, so we started a project a few years ago, and we're building a big, quite large advanced biologics manufacturing facility in Burnaby. This should be completed next year in 2022. So this was a $140 million investment. So stem cells providing $95 million of that. And then we got both federal and provincial support in the form of $45 million to help us with that. And so this is an advanced biologic manufacturing facility. So it's really a facility to make products under GMP for use in cell therapy. So you could use our media products in cell therapy or cell separation products in cell therapy. So this is going to make us a leader in these types of products. So it's been a great investment for us, and it's progressing along quite nicely. And I wanted to highlight that because I think this is kind of interesting given kind of what's going on right now with COVID. If you've been paying attention to the media and following it, there's a lot of talk right now about the need for Canada as a whole to increase its ability to do biomanufacturing. So its ability to manufacture diagnostics and therapeutics and vaccines. A lot of the shortages that we've been seeing with COVID in the supply of vaccines, for example, is because we don't have that manufacturing capacity. And so, Canada does need to build this infrastructure, but it's not enough just to, build the infrastructure to do the vaccine. You also have to have the supply of the kind of reagents that go into manufacturing the vaccine. So you need to have the enzymes to make the RNA. You need to be able to manufacture the RNA and so on. And so this is where stem cell is positioned quite well with this new facility to play an important role here. If you look at the components that go into diagnostic tests, or if you look at how you do the manufacturing for the therapeutics and the vaccines, these are the types of products that we sell. For the diagnostics, you need magnetic particles. You need to be able to make the illegal nucleotides that are used as the primer and probes. You need to make the enzymes that are used to do the amplification. And so we have all those products. And same thing with therapeutics. You need the media to culture the cells that the antibodies are expressed on and so forth. So we're very well positioned to play an important role in helping Canada secure that supply chain for these critical reagents. And so I'll just end with, things are going very well at the company. The life science sector is quite robust right now, so we're continuing to grow. Over the next 10 years, we're planning on investing about $1 billion in R&D, and we need to hire about 4,000 people. So if you're just graduating and you're interested in learning more about the company, feel free to reach out. I have the name of one of our recruiters, or you can just follow up with me during the network event. I'd be happy to speak with you. Thank you.
Good. Thank you so much, David, for your, as always, great presentation and letting us know all the great things that's going on at Stem Cell Technologies. And just to let everybody know, I just put a note in the chat that we will be extending the event till 5 o'clock, just so that we have time for some networking. So I'd now like to introduce Alison Orth, who will be introducing our next speaker, Dr. Gordon Wood. Alison holds a business degree from Simon Fraser University and is an ACRP certified clinical research professional. She holds more than 25 years of experience leading health research studies and project teams at both community and academic sites. Alison has facilitated over 80 industry and grant-funded clinical trials, outbreak investigations, retrospective chart reviews, attitudinal surveys, and evaluation projects. She has also held various research leadership positions in public and private sectors, including Fraser Health and the TASC Research Services, Inc. As part of her role as Unit Director with Clinical Trials BC, Alison recently led a national initiative to engage research participants about their clinical research experience to enhance patient and public participation in trials and improve their overall experience as a participant. So now over to you, Alison, to say a few words and introduce Dr. Gordon Wood.
Thank you so much, Ryan. Thank you to LifeSciences BC for inviting me here today and for hosting this showcase series. I always do my best to participate and go what used to be what we would call the road show to go around the province, but today we're doing that virtually. Clinical Trials BC, along with the BC Support Unit and Research Ethics BC, is part of the BC Academic Health Science Network. We work to advance BC's development as a world-class destination for clinical trials across the province's hospitals, research institutions, and communities. Our mission is to maximize the health, educational, and economic benefits of clinical trials to the citizens of BC. I am very honored to introduce our next speaker to you today. Vancouver Island is home to a significant number of clinical trials running at both hospital and community-based sites. And Dr. Gordon Wood is a site principal investigator for a number of trials focused on intensive and critical care. Dr. Wood is a Victoria native, currently practices as an anesthesiologist and intensive care medicine specialist in the Island Health Authority. Dr. Wood has belonged to the Canadian Clinical Trials Group since 1992 and has participated in dozens of academic and industry-sponsored multi-center clinical trials in ICU patients over the last 25 years. It's fantastic, Dr. Wood, and thank you so much for speaking to us today about the trials you're working on.
I want to talk about the critical care medicine research program that we have in the Island Health Authority. I'll talk a little bit about how I got into research in the first place, then about the program that we have in Victoria and Nanaimo now, and also a little bit about our involvement in research studies in COVID. So I was hired on at Queen's University in 1991 and to do critical care medicine. And I got there and they said, "Oh, Gordon, you can be the researcher." And I had no interest in research. I had no training in research, and nobody else wanted to do any. So I spent a couple of years doing a couple of unremarkable studies published in unremarkable journals and thought, well, there must be a better way to do this. And at that time, in 1989, a bunch of academic intensive care specialists who had interest in research formed the Canadian Critical Care Trials Group. So it was a consortium of academic trialists who produced an extremely collaborative trials group, and it became the most prolific collaborative critical care trials group in the world for many years. So my position was one of giving some input, but also mainly recruiting patients from Queen's University, from Kingston, Ontario, into the trials. And so the first study we published was one looking at two modes of trying to prevent stress ulcers in critically ill patients, which was a big problem. And our first paper went into the New England Journal, which for the kind of research I do is the world's most premier journal going. Everybody wants an article in the New England Journal, and I managed to get my name in there in the first go. And I thought, well, this is pretty cool, this research stuff. Certainly better than doing my own stuff that didn't make it very far. And the second study we did was called the TRICK trial, where up in the mid-90s, you had to have a hemoglobin of 100 as a patient if you were seriously ill. Physicians thought that having a hemoglobin any lower than that would add to your morbidity and mortality. But we did a trial that randomized patients to letting their hemoglobin go down to 70, which is about half a normal, versus pumping it up to over 100, and found that the patients actually had a better survival rate with the low hemoglobin. And this was basically revolutionary. It's been repeated many times, confirmed many times, and it's totally changed people's practice. And now physicians would never think about transfusing a patient with a hemoglobin of, say, 98. And so this was the second trial. And at this point, I was thinking, two New England Journal trials. Again, this is quite cool stuff to be doing. But I left Kingston and went to the United Kingdom to be the director of an intensive care unit there for a couple of years. Then I came back to Victoria, which is where my home is, worked in our community ICU, and missed the research I did. I missed the people that we met 3 times a year with to design these trials. I missed the meeting we had every year at Lake Louise, where we had great skiing. So I started to do group trials in Victoria. At that time, there was no research office. I had to find a research coordinator, Fiona Auld, who was an ICU nurse, to work with me. But we started to do industry and mainly trials groups, so academic trials. And I've brought on another physician, Dr. Daniel Overcombe, who's taking over for me as I sort of moved towards retirement. And in the last 15 to 18 years, we've participated in over 40 academic and industry-sponsored trials. Many of the publications have ended up in Tier 1 journals, so 4 or 5 articles in the New England Journal, a few in JAMA. So this is all very important research, and our job is to recruit patients. We were the first community hospital to participate in the Canadian Critical Care Trials Group. There's more now. And currently we are participating in 6 trials group studies and 1 industry trial. And we've also started doing 5 COVID trials, or we started doing that in the spring. Now there is a research and capacity office in Island Health at the Jubilee. It provides me with a lot of support, not only to pay the research coordinators from the funds we bring in from doing the trials, but they do all the ethics and all the submissions and all the regulatory work that a clinician like me doesn't really want to do. We've also expanded into Nanaimo. We're doing some of our trials group studies up in Nanaimo, having got a couple of physicians up there interested in it. So in 2009, H1N1 blew through. We had a little bit of a warning because it hit the Southern Hemisphere first, but when it came time to start to try to do trials on H1N1 therapy, the whole system was too slow. The pandemic passed us by before we got our first study off the ground. So the clinical trials groups across the world, but in Canada too, vowed not to let that happen again. And this time, when COVID came, we were ready to have expedited research studies available, and so they could get off the ground while the clinical problem was still there. Victoria has been hampered a little bit by the fact that we didn't get a lot of COVID patients, which was in some ways good. We probably got less COVID patients overall than we got offered studies to do on COVID patients. So we had to pick only the ones that we thought that we would be able to recruit patients into. The first study was a Sprint-SARI. It was a worldwide study. It was a prospective observational study in all COVID positive patients admitted to the hospitals. So it was trying to figure out what type of patient came in, how they were treated, what their outcomes were, and we enrolled 33 patients in Nanaimo and Victoria. We continue to participate in CATCO. Across the world, this is called Solidarity, but in Canada it's called CATCO, and it's what's called an adaptive study. These adaptive studies came out of the failure to develop anything useful during H1N1. Because of the time it took to get things going. So you create one placebo group, and then you can add different investigational therapies that can come in and out of the trial. So we were studying initially placebo versus hydroxychloroquine. We also used lopinavir, ritonavir, which were AIDS drugs from the 1990s. Some centers across the world were looking at interferon as well. And we didn't start with remdesivir right away because we didn't have any supplies, but eventually we got supplies and we were using that in one arm as well. As these therapies were shown to be non-efficacious, like hydroxychloroquine, they were dropped. And the AIDS drug as well, interferon, was shown not to be useful. And now we're left with just remdesivir. In all, we enrolled 24 patients in Nanaimo and Victoria. We did CONCOR, which was convalescent plasma. So you take the plasma from patients who have had COVID, hoping there's antibodies in there that if you transfuse into other patients, will improve the outcome of that patient. Unfortunately, this therapy didn't work, not just in our study. There are other convalescent studies around the world. We enrolled 2 patients in Victoria, and then it was shut down due to futility. Another study we did was ATTAC. There was concern that in COVID, a lot of the morbidity and mortality was due to thrombosis, so strokes, heart attacks, DVTs, pulmonary emboli. And it compared therapeutic doses of a low molecular weight heparin, enoxaparin, to prophylactic doses. So therapeutic doses are used if somebody has a pulmonary embolism, and prophylactic doses are used to prevent things like deep vein thrombosis and pulmonary emboli. And interestingly, this therapeutic dose of enoxaparin benefited moderately ill patients on the ward, and it was stopped for efficacy. and it had no benefit in ICU patients, and it was stopped due to futility. And we put 2 patients in that study as well. And at the present time, we're doing a study looking at angiotensin receptor blockers. The COVID virus gets into the cells by binding to the angiotensin receptors, and if you have a drug blocking that, maybe that will reduce the morbidity and mortality from COVID. We're just getting going on that, although I think the COVID patients coming into the hospital in the ICU is going to continue to fall. So in summary, I've spent 30 years doing recruiting patients into studies, mainly for the Canadian Critical Care Trials Group. I'm not really a researcher, but in my spare time, I've done this, and I found it very rewarding. You stay into contact with other people in the country. There's no money in it for anybody who wants to get in on it, but there's lots of an opportunity. We have a research office in Victoria, and any clinicians who would like to bring clinical trials from elsewhere to Victoria can get the research office to help them supply all the administrative structure that you need to do a study, so you can just concentrate on recruiting patients. So that's really all I have to say. Thanks again for inviting me.
Thank you very much, Dr. Wood. That was a great presentation. So we thank you very much. And now on to our next speaker. So our next speaker is Ann Woods, who is the Managing Director of the Life Science and Healthcare Practice for Silicon Valley Bank in Toronto, whereas she is responsible for leading the bank's efforts in this sector across Canada. Ann has close to 25 years experience in capital markets and life sciences. She began her career as a financial analyst, and in 2005 went back to her roots in life sciences. Prior to joining SBB, Ann spent several years as a director with the Pagea Group, bringing insights and strategic direction to life science and health care clients. In 2018, Ann joined the board of Life Sciences Ontario. She holds a BSc from McGill University and an MA from the University of Guelph. She is also a CFA chart holder. Ann is a passionate life sciences leader who generously volunteers a great deal of her time to supporting the life sciences ecosystem. And especially I can say that with lots of time with LifeSciences BC, Ann has been very generous for their time, and we greatly appreciate it.
Okay, hi everybody. Thanks so much for sticking with us through all of our technical issues. As Ryan said, I lead the life science and healthcare practice for Silicon Valley Bank in Canada. So I should always ask to go first, because we are really the boring but essential part of research and development. We're the commercial bank to the innovation economy, really, and a big part of that is our work with life science and healthcare companies. So we have over 3,000 companies that work with us, and when you look at the sort of best and brightest of these companies, if you take just a group of companies that have gone public in the last 3 years, 70% of them actually bank with SVB, and what that really means is we have access to people and investors and insights that give us some really interesting information that we pull together into reports a number of times a year. So the data that I'm going to run through today comes from those reports, and all of that's available on our website. So I'm just going to dive in a little bit on what happened in 2020. Talk about some trends that we see for 2021, and then what I think we need to do in Canada to make sure that we kind of continue to ride this momentum. So suffice it to say that 2020 was a year of contradictions for sure. To say it was interesting would be a bit of an understatement. Global lockdowns resulted in 12.5 trillion of global sort of shortfall in output, and governments really had to dig deep into their coffers to support the economy. But at the same time, what we saw was record fundraising and record amounts of money raised by companies across all sectors in the innovation economy. And then we saw markets really significantly outperform expectations. And so I think, really COVID really kind of drove the importance of innovation to the forefront. And I think no more so than in our sector, where innovation is not only going to help us get out of this mess, but it's also going to provide that foundation for future economic growth. And so if you think about, some data that will help sort of, prove that, I'm not alone in my bullishness, you can see that, we are in the midst of a sort of, a biotech boom that's lasted for many years. But what we saw in 2020 was fully, 16.8 billion was raised by US venture capital firms. So just in the US alone, $16.8 billion went into funds. Money is going to be looking for a home in new and innovative companies. And so in addition to healthcare funds raising more money, the one thing that we see is sort of typical sort of generalist investors. So investors that never really looked at the sector before are really starting to take an interest. And so that's driving some of that additional capital as well. And so, when we see all this money that went into funds, what did that mean for actual companies? And so again, we saw 2020 was a record year. And this time last year, we produced a report that said we continue to be bullish on the sector because good technology is good technology. But I don't think even we could have predicted what really a number of deals that would have happened. And so, if you think about the third quarter of this year, we were predicting that people would, third quarter of last year, we were predicting that people would not be comfortable doing deals over Zoom. And instead, what we saw was the third quarter being actually the largest quarter in history for both number of deals and dollars that were invested. And so these two slides give us a good taste of what's happening in the US and Europe, but how have things played out in Canada? Has Canada sort of participated in this biotech boom? And so if you've been following the news at all, you'll know that Canadian companies really are, had a great year. And what we saw is the three largest biotech IPOs in Canadian history with Repair, Fusion, and Accelera. And so Canada did have absolutely a record year in both dollars and number of deals. If we look at both public and private dollars and deals that were invested, there was 57 deals and over $2 billion went into life science companies in Canada. And I think for me, what is most promising is when we look at the early stage deals, there was a time where we really felt like there was this sort of valley of death where companies couldn't attract capital to get them to kind of scale up. And now what we're seeing is, in fact, when we look at early stage deals, the median deal size for a Canadian company is actually bigger than the US and Europe. Now, that is probably has something to do with the fact that our overall ecosystem is skewed a little bit more towards biopharma, which typically has bigger deals. But what I like about this is Canada has for a long time had a reputation of not raising enough capital to get their early stage companies sort of on the right trajectory. And this says that maybe that's not the case anymore. And then, of course, I had to highlight some of BC's success stories because BC really is the darling of Canada when it comes to life sciences. So life sciences is the fastest growing sector in BC, both in terms of company creation and employees. The sector is growing faster than both Ontario and Quebec. And the sector attracts an outsized amount of venture capital. So when you look at all the venture capital dollars invested in BC, 40% of them go towards life science companies, and that number is only 15% when you look at the rest of the country. So, all of this really leaves us feeling really bullish about the sort of global life science ecosystem. And we continue to think that Canada's growth rate is going to exceed the growth rate both in the United States and in Europe. But in order for that to be true, we need to really take a few things into account. And so there's been a lot of conversation about how we can maintain this sort of momentum and Canada continue to succeed. And I participated in a number of conversations, and I sort of took everything that I've heard and kind of boiled it down into three categories of things I think we need in order to be successful. So the first one is really important, mindset. So we're seeing a big mindset shift from 10 years ago where intellectual property and technology was developed and then very quickly sold off for somebody else to commercialize it. Now there's a real track record of success for Canadian companies where young entrepreneurs have sort of a vision that they can sort of hold on to and say, look, this is what I can be. And I think that's really emboldening them with the courage to embark and really risk, really risk taking a chance and growing their companies at home. And I think once you get into that mindset shift, now you have to start looking at policy changes. So one of the things that we've talked a lot about is intellectual property and moving that out of Canada. And the reason that's done is for tax purposes. And so if we had globally competitive intellectual property policies, not only could we keep our IP at home, but we could probably attract IP to Canada as well. And I think another piece around policy that's important is, in many ways, big pharma has been vilified for various reasons, but drug prices have been the one that has really been very high on the list of policy changes. And I think we're seeing a focus on drug prices is pretty short-sighted. And perhaps the vaccine rollout in the US versus Canada is providing us with some evidence of what that short-sightedness leads to. And then, of course, we talk about capital. And usually when we talk about capital, we mean money. And it is absolutely important that we get more domestic investment into the sector. But I think I'm thinking of capital more holistically. So, of course, people and talent. So, we have a little bit of a virtuous cycle happening here where now we've got a track record of talent that has had success that can now move on to new companies and continue that success. And so I think we need to make sure that we continue building that virtuous cycle. We talked about a little bit about intellectual property, and then I think Dave Llewellyn, he mentioned this piece around kind of capital assets and real estate and infrastructure, and that's something that's becoming more and more obvious because we've got the mindset, we've got the money, we've got the people. Now we have more science than we can actually develop, and so if we can get the infrastructure in place that are sort of turnkey solutions, I think that's going to be really, really impactful, and it's going to stop companies from saying, look, I can move to Boston or I can move to the Bay Area to develop my company. So all of these things, of course, are very connected, but I think if we get the mindset and the policy and the capital all in place, we're destined to be a top 3 player in life sciences, and so I'm really looking forward to being a part of that. And so with that, I'll hand it back to the LSBC team.
Thank you, Anne, and always a great presentation. And I'll also highlight for our audience that Silicon Valley Bank always has really interesting reports. So we always encourage you to keep an eye on the LifeSciences BC newsletters. We will feature them there, and also checking out their website. So thank you again, Anne, and also for being a great sponsor of the series. So our final speaker for today is Sam Mercer, who's the principal for Samuel Mercer Consulting and also the board chairperson for Vancouver Island Life Sciences. So Sam has been a great partner of LifeSciences BC. He has over 20 years of pharmaceutical development experience, previously managing external agreements and outsourcing in the areas of global clinical trials, IT and scientific consulting, academic alliances, and pharma licensing, having negotiated directly over $600 million US in varied external agreements for both Roche and Novartis in Basel, Switzerland. Prior to this, he managed proposals, contracts, and business development at several global organizations in the UK and US. And more recently, he is a management consultant, interim executive, board member, and advisor to a number of biotechnology and software tech startups. His focus is on the technology and IP development, clinical trials strategy, startup community strategy and funding, and negotiating and managing of complex business agreements. And also would like to say that Sam was a wonderful entrepreneur in residence for our recently completed investor readiness program. So he was a great asset to that program as well. And so without further ado, I'll turn it over to Sam to give a few remarks.
So thanks everyone. And I'm standing between you. Normally you'd be stampeding me, trampling me on your way to the bar as we wrap up the event, but luckily a little bit of the stress is off because I'm only holding you back though, but I am holding you back from the networking table, so I'll let us get to that. So thanks for all the great talks, and as always, really good content. I'm Sam Mercer. I chair the nonprofit VILS. We also have a great board of 6 other people, many of whom are here in this session. I've seen you in there. In that last event at the Delta, that was the last VILS in-person event as well. And I still cringe when I think about, my ignorance and thinking it was all about fomites and just thinking we could just wash our hands a lot, and there we were all speaking moistly in each other's faces, but thinking that if we just washed our hands a lot that we'd be fine. So I cringe a bit. At that last event, I made the mistake of not fully introducing VILS to those who are less familiar. So this time, I know there are some on here who need a little introduction to VILS. So what is VILS? It's Vancouver Island Life Sciences, VILS.ca. It's not only biopharma industry by a long shot, it's, UBIC, Camosun, VIU, we obviously actually need to build with. Dr. Dames and your colleagues, local health authorities, startups, consultants, service providers. As Ryan said, we think we've always been a good friend, a great friend and sister organization to LSBC. But why? So then why another group on the island? Why Victoria? Well, the island and Victoria are really active and increasingly so, but we're physically separate. And Canada is pretty bad for that. It's a major issue all across Canada, but BC and the island were even more physically disconnected. So we strengthen that connective tissue between groups. And also to David Llewellyn's point earlier, we need to strengthen our regional infrastructure, including biomanufacturing. We've got a $1 billion overdue sewage plant here in Victoria now, so there's no reason we can't spend a fifth of that, $200 million, on a cell-based manufacturing plant here in town. And to Anne's point earlier, we found the real estate counterintuitively for that $1 billion sewage plant on military land. So again, no reason we couldn't find $200 million worth of space for cell-based manufacturing. So the Victoria community as well, it's special. It's a little different. In the island community, we've got a particularly wide focus by necessity, a big tent, as I like to call it, and we're into the convergence of technology areas. We're strong here in software. We'd like to be stronger in mental health. We have what Carl Hansen on an LSBC event the other day called an irreverence for traditional boundaries between disciplines and technologies. We think that's part of our culture. And psychedelics, as Dr. Dames presented, are a case in point, and kudos for that. It takes a lot of fortitude and courage to work in that area in the face of a lot of historical bias. And the island can lead in that area. And there's still a lot of issues to fix here just on the island. Like there's still an opioid epidemic going on. There are, of course, some surprisingly outsized life science players that were on Anne's slide, like Aurinia, 1.75 billion US market cap today. Immunoprecise Antibodies has a 220 million market cap, and Eupraxia Pharmaceuticals, who I had the fun and privilege to help build from the beginning, and not everyone even knows. It should be bigger news. They're public just a few days ago, sort of a 120 million Canadian market cap, depending on who you ask. And now Oak Bay Bio, David just presented earlier and has a round open. So now's the time to get in on that opportunity. Almost done. Since 2016, VILS has held open, free, and fun and unpretentious events, always in a new location, and pre-pandemic every month. We often had a tour and talk format in different places, which people really like. And how were we able to do that? Well, as Oak Bay Bio exemplifies, there is a new company, not every month, but almost, in Victoria. And then you'd say, well, how is it possible to do all that and be free and be run by all volunteers like our board and myself? Well, we think we proved we can do a lot with almost no budget, but I would say watch this space, because we think soon we'll be doing a lot more without losing any of our grassroots feel. And so finally, I'm going to paste some links into the chat, so there's no excuse for not signing up for the mailing list, because that's one question. How can you engage with us? Well, you can join the mailing list at vils.ca. You can follow us on LinkedIn, and you can reach out by email and phone, and I'll put my details in here. And I welcome us going to chat. And it works pretty well in this system, so do hang around to chat at the tables. Thank you. Thank you for having me.
Perfect. Thanks you so much, Sam. And, we really look forward to being back on the island in person. And, hard to believe a year ago we were touring all around all the great companies there. So in closing, I'd really like to thank all of our speakers today, Dr. David Kroger, Dr. Shannon Dames, Dr. David Goodlett, Dr. David Llewellyn, Dr. Gordon Woods, and Sam Mercer. And this showcase series is a program where LifeSciences BC, with our partners, travel to different locations throughout British Columbia to showcase life sciences and research and innovation. This series is only possible due to the generous support of our partners. So today we had our event sponsors, Silicon Valley Bank, Stem Cell Technologies, our speaker supporters, Clinical Trials BC, and the Michael Smith Foundation for Health Research, and our event supporter, the University of Victoria. I really appreciate all of our speakers for coming today and the wonderful presentations and bearing with us. And to all of you as our attendees for really coming and offering great insights and participating in the chat and all that good stuff. So coming up next, April 14th, we have a Lab to Patient Summit. So that's going to be a great event. More details to come on that. May 4th, we've got the Blake's Breakfast event. That's going to feature James Taylor of Precision Nanosystems. So that's going to be a fantastic event. The showcase series continues on May the 13th. We're out to Surrey, and May 20th, we've got an international trade event, which we're quite looking forward to. And so now, for about the next 20 minutes or so, we're going to go into networking, which I know that you will all enjoy. And the very last slide we have, this is all of our sponsors, and so of the organization. So you can see all of our platinum, gold, silver, and bronze sponsors, who without support and all of our members, we would not be able to continue all the work that we do, as we do not receive any government funding. So on behalf of Wendy Herbert, our President and CEO, all of our speakers, all of our staff, and thank you very much. And also a special thanks to Tim Hennessy, who is our manager of events, for all his behind the scenes and all that good stuff. And we are now going to go into the networking session. So thank you again. I'll look forward to seeing you at the tables then. Thank you.